Henry is the scientific director of the Institute on Stem Cell Research, Brain Research in Lyon. And one reason why I always love to get Henry here, because is that Henry has this very unique profile in many different respects, but I will only emphasize one, which is that he has been very systematic looking at the anatomical organization of the brain, in particular the neocortex, even though we might hear about some other structures today. Let's see what kind of surprises we're going to have. And nowadays in the field of neuroscience, these are like becoming very rare exceptions, that people are really sort of chipping away at the details of the wiring and the connectivity, which I do believe actually are going to give us important insights, as also saw in John's talk and Leah's talk, important insights in the organization of brains, and also then extraction of principles that will feed into the technologies that we might want to build to study these same principles. So Henry, great to have you here, and I'm looking forward to your talk.
Thank you. Thank you very much for your invitation. Actually, I'm no longer the director of the Institute. It's Colette Dehay.
She's scientific director.
Oh, well, I participate in that, I guess. So this is where I come from. This is Lyon. It's a very nice town for those of you who haven't been there. It's not maybe quite as active at night as Barcelona seems to be, or as Barcelona was the last time I was here. But yeah, centre of Lyon. How do I change slides? Is there a...
Okay, this remote, is this remote on? Okay, there's a magic button somewhere.
The magic button.
There you go. Here.
And how do I go back?
This left is back and right is forward.
Okay.
So now you go back.
Yep.
Right is forward.
Okay, so if you just want to remember one thing from this talk, the title could be very usefully The Operation of a Distance Rule and the Shaping of Connectivity in Primate Cortex. Primate cortex because you're allowed to do track tracing, you're allowed to do the sort of anatomy that I'm going to be talking about in primate cortex, non-human primate cortex. But basically what I'm going to be telling you is that there's an operation of the weight of connections over distance, which gives an insight into the organizational principles of the cortex, which I believe are very useful. That's what I'll be putting forward. Another title, the title I actually gave, is The Brain in Space, and I'll come round to why I've given it this title at the end. Now, over the last 10, 15 years, there's been a number of people looking at what they refer to as large-scale models of cortical organization, architecture, the architecture of the connectivity between, and I'm going to be talking about cortical areas. And this has led to a huge number of publications dealing with a model of the cortex which is a small world. The notion of a small world network comes from social studies where people have been very interested in the organizational principles between individuals in a society, because you want to understand rumor propagation, you want to understand disease propagation, you want to understand the actual interactions between individuals. And from these studies in the 60s came the notion that the social organization of the human race is based on a small world network. The small world network refers to the notion that you have a low number of degrees, that's to say hops, going from one individual to another, and it might be as few as 6 or 7, which separates any individual in this room from any other individual on the planet. So you're 6 handshakes from the Queen of England, which is reassuring, and maybe a dozen or so from Hitler, which might be less reassuring. Now, this led to an enormous number of publications with small world in them. And I think a lot of people probably didn't read those publications, because the argument I'm going to be putting forward is actually understanding the cortical network, i.e. the areas of the cortex and the connections between them, in terms of it being a small world, is actually not very useful. That's an argument I'm going to be putting forward. And to to be able to get that to that point, we've been doing a large number of anatomical investigations using retrograde tracers to determine the connections between cortical areas. And this has been done by numerous scientists, including John Cass and Leah Krubitzer and many other people. The only thing that we brought to the table, which was a little different, is that we were very systematic in the way we did that. And we counted every connection. And so that meant we found a lot of very, very weak connections. And so the data I'm going to present to you includes a database with many weak connections. I think that the temptation is to forget about the weak connections. And to this day, I really don't know if weak connections are that important. But coming back to the social sciences, this is probably the most cited paper, far more than small world networks, which is the strength of the weak tie. Mark Granvetter, who I think is still operating, was very interested in what are the factors which lead to social cohesion. What is it that which enables an integration of individuals into the social fabric? And the example that he gives is, in fact, that you actually get your future job or you meet your future wife or something through some sort of chance encounter through a weak connection. And so this notion of the strength of the weak connection is that the weak connection gives you a very high integrative power in the network. So there you come back to exactly what the small world is banging on about. The small world is interesting for the neuroscientist because it's all to do with high integration with economy of connectivity. And if you imagine whatever it is the brain is doing, that would appear to be rather appealing. So there's a sort of social science aspect to what I'm going to be talking about, because the way we got into this small world conundrum was very much to do with the approach which had been adopted, which was very much taking a database which had been collated across studies and using that database to try and understand the global organization of the cortex. And that turned out to have a lot of problems attached to it, because the way we segment the brain into different cortical areas can differ between groups, the sorts of tracers you're using can differ, and the attention that you're giving to the connections can be very different. In other words, if you're studying the visual system and you're interested in the connectivity of area V1, you might ignore the fact that, yes, there are projections from way beyond the visual system into area V1 that you might not be taking any notice of. So that the studies had been done by various anatomists who were interested in the auditory system, the somatosensory system, the visual system, and were perhaps less interested in connections that fell outside of their immediate interest. And so what's interesting about this is, and I'll come back onto that, is as I'm saying, there's a sort of social science thing where people have taken on board very imperfect databases, imperfect in the sense that they're very incomplete, to make large-scale models of the cortex, and that is part of the problem. So I don't know how long this goes on for. Is it an hour and a half? Is it an hour? Okay, well, we'll see how far we get. But actually, if you're talking to neurophysiologists, they're usually not interested in anatomy at all. They believe that function doesn't depend in any way on structure, which is a kind of mystical standpoint, if you think about it. And they're not interested in these large-scale models of the cortex, really. I mean, they see all these papers with small world this and small world that, what have you. But what they are interested in is a laminar pattern of connections between cortical areas. And this has enabled people to think of cortical areas as being arranged in some sort of hierarchical process. And that is much more interesting to a large number of physiologists, and I have some findings which are relevant to that, and if we have time, I'll talk about that. So, and I hope at the end of it to be able to explain why the brain in space has any kind of any kind of interest. So I mentioned that the database that people have been using was very incomplete. And it was very incomplete in a very simple sense. If you do a number of, if you're looking at the connectivity of the cortex generally, you make a square matrix. So you're interested in areas 1 to 20, and you arrange them like that. You see these patterns in journals all the time now. And then you have 1 to 20 here and 1 to 20 there. And when you have a black patch, those two areas are connected. That's a square matrix. That means the status of every connection that you're talking about is known. It either exists or it doesn't exist. And I will argue that you need to know the strength of that connection. Why? Well, it turns out that the density of those matrices, that is to say, the percentage of possible connections that can exist, is actually very, very high. It's around about 70%. So 100% is every area is connected to every other area. So coming back to the small world notion, what's interesting about the human race in 6 degrees is the human race is billions, and 6 degrees is a very small number. That's what's intriguing, that's what's interesting, and that's what captured the interest of a number of anatomists and physiologists at the beginning of this century. So if you take a room at the end of this week, presumably when you've been to enough bars and restaurants and you've been up all night eating tapas and what have you, you all know each other. The density of this audience would then be, in terms of connectivity, 100%. Each person knows each person. And so it's a small world, but it's trivial. It's simply a small world because you all are connected. So at 70%, it's not really very interesting, it's not very exciting to think about the inter-aerial network as being a small world. So that's the point, that's the first point I'm going to be talking about, exit small world because of the density. The second point I want to make, and I won't go into it with any kind of data slides, but you probably have heard about this, is there's a big interest now in hubs. When you talk to network scientists, they love hubs. And the reason why they love hubs is because they route information. This notion of routing information is, so you have a sort of switching process, which is absolutely extraordinary. It's really taking the telephone system to heart. And it's imagining that the brain works in a system of routers that switch and switch off and switch in and what have you. And when you do that, the claim is that you have hubs, just like you have in Barcelona is a hub. So if you want to go from here to Chicago, you have planes that go straight from one hub to the next hub. That's why it's a hub. But if you want to go to some little town in Spain, you come to Barcelona, then you get onto the local plane, and you eventually get to this small place you want to get to. So Barcelona is a hub. So this idea of the importance of hubs in the brain and the switching and the routing, and you might notice I'm being a little sort of flippant about that, is that the hubs are over-connected to what you would predict statistically. That the probability of connections between hubs is greater than you would predict. This goes back to a notion which is referred to as the rich club. Rich clubs are organizations of capitalists who get onto the governing board of every enterprise of any importance. And then at night, these people meet, they smoke cigars and they drink fine whiskies, and they make decisions. And those decisions rule the economy because these guys are each individually in very important positions. This is the notion of the rich club that has been developed. And so the rich club in the brain would be a network of hubs which are more interconnected than you would expect by chance. So they have a high in degree, they have large numbers of connections, but they're more connected amongst themselves than you would predict from that high connectivity. And that would, of course, put these hubs into a position of control, if that's what you're going to believe. Well, I won't go into that, but if you think about it, if you have a high density of connections, there's a problem with the notion of small world, okay? Because if it's a density of 80 or 90%, to talk about this being a small world group in this room doesn't really make very much sense. But if you have a high density, it also gets very, very difficult mathematically to define these rich clubs. So next time you hear about rich clubs, remember, there's a problem there if you really take the data seriously.
Henry, for clarification, is it really a fair criticism, you could argue? Because in the literature, when people talk about small worlds and rich clubs and so on, often what they talk about is a functional connectivity, which in the end is some statistical pattern in signals people extract from the brain, like an fMRI signal. You say, okay, this is my correlation matrix, right? And they could now argue, okay, but I'm telling you something about functional connectivity in terms of the response properties of the system. I'm not telling you anything about physical connections.
To this. Functional connectivity is the worst terminology that anybody has ever thought of.
Right,
it should be removed from the dictionary. Let me just tell you what functional connectivity is. First of all, it doesn't necessarily have anything to do with connectivity, point one. And secondly, it doesn't necessarily have anything to do with function. And you put those two terms together, and you've really squared the circle. So what is functional connectivity? Functional connectivity is observing that different parts of the brain have some kind of synchronous activity, that there's something simultaneous going on in different places. That can be caused because there's actually a physical connection between those areas, or they can be common input, or they can be all combinations of those things. So actually functional connectivity, what people doing functional connectivity do with the small world issue, is they say, oh, we have a small world in terms of functional connectivity, and then they go one step further and they say, if we look at the anatomical connectivity, we have an underlying connectivity of connections, physical connections, which supports the functional connectivity we've been looking at. But I really think we should distinguish the two things and leave them, you do one or you do the other, but bringing them in together, I think, is a confusion. And it's a terrible name. There's no doubt. Even Olaf Sporns, who I think actually coined the term, admits that it's an abomination. It really is not credit to the field. So the technology I'm using is, it's very, very, very old-fashioned. You inject a tracer in a target area. The tracer is retrogradely transported to the soma. It takes about 10 days. You sacrifice the animal, and then you look at where you have cell bodies in the brain. And where you have cell bodies in the brain, you can deduce that you have a projection from that neuron, in wherever that neuron is, in an area or in a subcortical structure, to the target area. The tracers are very sensitive. They're very sensitive. They do not show anterior grade transport, and they're not carried across synapses. So what you're seeing is what you're getting. The other advantage is that the injection sites are very localized, so you have a pretty good idea about where the tracer's coming. Using these tracers, if you make an injection in area V1 is the primary visual area. It's a huge area. Thousands of people have looked at this, the connectivity of area V1. It's been a well-studied area. It's a very large area in non-human primates. And this slide is color-coded. And the only point I want to make here is that if you make this injection in area V1 in 2011, when anatomy had already gone out of fashion, because people were writing journal articles saying that we now know about the structure of the brain, and so we can now do the important thing, which is find out about its function, you could still find projections which hadn't been then reported. And in fact, it was that kind of finding that earlier in the century that we noticed that there was a projection from the auditory cortex to the peripheral part of area V1 in 2005, which sort of woke one up to the fact that actually perhaps we didn't know the connectivity of the brain as well as we thought we did. So my subsequent slides are color-coded. And this is simply to show you that so they're color-coded, the known connections, a sandy yellow, and the connections which had not previously been reported are bright red, and the vertical axis is the number of areas, and these are 29 injections. The claim is that they're evenly distributed across the cortex. That's a bold claim. There's some holes in that one, but for the sake of argument, let's say they're evenly distributed across the cortex. And we're reporting here the number of connections which actually hadn't been reported previously to 2012. And here the axis is the strength of the connections, and here the axis is the distance from the area that was injected. So if you look at the nearby connections, they're mostly known. Within 5 millimeters or so, most of the connections are known. And as you go further and further away from the injection site, the proportion of previously not reported connections increases. So frequently people ask me, well, why didn't people report these connections? And one answer is because they tend to be further away. And if you're interested in the visual system, you look at the visual cortex. And if you're interested in the auditory system, you look at the auditory cortex. And visual areas are grouped together, and auditory areas are grouped together, so you tend to not cover a huge amount of distance. Over here, this shows you that the strongest connections are entirely known, and as the connections get weaker, the proportion of previously not reported connections increases. So I said at the beginning that yes, I'm talking about a lot of weak connections, but notice that, I mean, overall, the range of previously reported and unreported connections overlap reasonably well. So these weak connections I'm talking about are not, amazingly weak. They're not impossibly weak. Sorry.
Just for clarification, how do you define strength? Just the number of connections, or can you also say something on the strength of the single connections?
We'll come to that later. I'm not fobbing you off because that's actually going to be the heart of what I'm really going to be saying here.
This is all interview one, right?
No, this is 29 areas across the cortex. So these are some hundreds of projections to tens of areas. So it's representative. The claim is that this is very representative.
And later you also tell us about laminar distribution.
Yep, that will also come in later on. So now I'm going to be talking about the density of connections. And the reason why the strength of connection is interesting, because if you have this very high density, if everything connects to everything else, then the specificity in the connectivity is going to be not in whether it exists or not. It's going to be the strength of the connection. That's going to be the underlying message. But here I'm going to talk about the psychology or the sociology or what have you of the small world. What I've presented here is this is the graph density. So the density of the graph that I'm reporting, the network I'm reporting here, is 67%. So 67% is the percentage of connections that can exist and do exist. 100% is being the maximum number of connections, everything connected to everything. In fact, it's 67%, so 2/3. That's a very, very high number. And here we randomly remove connections, beginning with the weakest connections. And as you do that, your path length, so at this density, it's a little under 1.5 hops from any area to any other area. So you have a highly dense, connected, interconnected network of areas. And as you randomly remove connections, your average path length goes from a little under 1.5 up to about 3. And at this point, the graph begins to disintegrate. What does that mean? It means that you then have areas that you can no longer get to. You can't get to them from any other area, and so the graph is no longer a complete graph. And superimposed on that, I'm now going to give you the databases. So this is our database. This is the density we reported, and these are the densities that other people reported. So the classical study here is Feldman and Van Essen in 1991. Feldman and Van Essen in 1991 did a parametric study across, 400 or 500 papers, where they were able to put together a large database which went in the direction of this hierarchy of connections, and I'm going to come to at the end of my talk. But that database has been hugely used by people interested in modeling the cortex as a database, and people have added on to that. So they had a database in 1991, which was a little bit over 30%. And they predicted that if they had looked at the non-existing connections, the connections, that had not been tested, they predicted that they would have a density of 45%. All of these studies behind Feldman and Van Essen used various updates. I'll come to you in a minute. Yes. All the studies left of this are using the David Feldman and Van Essen, and they're all arguing in favor of a small world. And what they've done is they've taken the Feldman and Van Essen, and they've taken that database, and if the connection hasn't been tested, they say it doesn't exist. That's not what David Van Essen did. David Van Essen said, at a guess, if you tested these connections, I think they would be 45%. That's what his guess was. Now, in 1998, a guy called Jouve, who was a postdoc of Michel Imbert, took the David Van Essen database, 1998, updated it with the connections which have subsequently been reported, and used an inference method using second-order relationships to best guess what the density would be. And he came up with 58%. So that was a sort of reasonable process to say, well, we don't know if this area is connected, but let's use second-order relationships. So if A is connected to B, and we don't know if B is connected to C, and if A is connected to the partners of B, then probably the connection from B to C will exist. That kind of inference. And his prediction was 58%. So it's not very different from what we're actually finding at 67%. So these studies here, Honey is a 2007 PNS paper which claims that the cortical organization is a small world. So the small world, that's this notion that there's a very small number of hops going from one area to the next. And because they've ignored the non-tested connections, the density that they're working with is 20%, around 22%. At 22%, that kind of density, to say that there's a small number of hops going from any one area to any other, and therefore it's a small world organization, is pretty reasonable. You could make that. Malcolm Young, who did a lot of work on David Van Essen's database, who also made exactly the same claim, in 1993 had a density of 15%. So that density is by adding on new areas that people have reported. But again, as you add more areas in, the untested areas increases and your density plummets, okay? 15%. This is a recent, very, very highly cited paper from Moder and Singe. It's also in PNS. It's 2010. And they've done exactly the same thing. They've added in more and more areas, and actually definitions of areas which makes the mind boggle, the boggling mind. And they end up with a density which is around about 7%. And in their graph, there's 14 areas which are unreachable. So they actually don't have a complete graph. You can't go from, there's 14 areas that you can't reach from any other area that are unconnected. There you are, unconnected. Small world. So that's a very strong claim. Now, there's light at the end of the tunnel, because the people who started this small world bonanza is, the people who started this small world bonanza is Watson Strogatz, who came up with a process which would enable you to decide if you're actually dealing with a small world network or not. They observed that if you take a lattice, a regular lattice, this is a regular lattice where each area is connected to its adjacent area, and you randomly rewire that progressively. As you do this random rewiring, there's a rapid decrease in the path length. So the number of hops required in this lattice to go from here to there is quite straightforward. You've got to go through every single dot that's on this lattice. As you introduce these rewiring process, you're going to make shortcuts. As the shortcuts increase, your path length is going to decrease until you get to a fully random network. While you do that, there's a measure called the clustering index, which interestingly does not change. It remains very high. What is the clustering index? The clustering index is your friends usually know each other. The reason why the human race has a small world network is actually because there's a few cuts across the society. So you will have some acquaintance who is in a different neighborhood, and in other words, a different connectivity neighborhood. He will have a different set of friends, and his friends will know each other. So with very, very few cuts across, because of this high clustering, the high clustering index, the friends knowing friends effect, this is where the small world phenomenon is going to happen. So the small world, according to Watson and Strogatz, is this region here. At this region here, you have a short path length and you have a high clustering. If you then continue this random wiring process, then the clustering plummets and you get here to a random network. At a random network, there's no clustering and there's a very short path length. That's the definition of a random network. So what we're doing here now is simply playing in this is a very, very low density of around about 5%. If you remember, this is the Modder and Singe PNS paper I mentioned to you. They had a 5% density. They were claiming it was a small world. And I said, well, it makes sense. You have a low density. They were working at a density like this, 5%, and you have this small world interval, if you like. And as you increase this density, these are the changes in the path lengths, and you get to a point where the rewiring will have absolutely no effect. So, Tony, I can't remember the name of the chap in your lab who did this, but this actually comes back from him. Gurney?
Mark Humphreys?
Sorry?
Mark Humphreys.
Humphreys and Gurney, is it?
Kevin Gurney?
Yeah.
Kevin Gurney.
The three of us did it.
Right. So this comes back to the approach that they had. So I said there's hope at the end of the tunnel because all you need to do with this approach, which was developed by Tony's group, is rewire your network. And if your clustering index changes, then yes, you might have a small world network. But the point I want to make is that we got into this conundrum at the beginning because of the willingness for people modeling the cortex to ignore the fact that they were using incomplete data and doing this amazing thing. You know, it's a bit like in the days of Joseph Stalin, you would sort of paint people out of the picture. You know, you'd have a photograph of a group of people, and then you'd knock somebody off.
In North Korea, they still do that.
And so you knock somebody off, and then you just paint him out of the picture, and he doesn't exist. So there's a sort of a kind of like tempering with the data, which is a bit terrifying. So so it is a very, very high density, and what does this mean? Well, it in a sense, this is looking at the distance from the injection site, from the target area. So there's 29 areas, okay? So this is a pattern which goes across the cortex, okay? And you're looking at the percentage of areas which are actually connected. So at short distances, everything is connected to everything. That means that neighboring areas are connected. So if I say to you, I've got area X, and around it there's Y, Z, and W, and they project to it, you say, well, I know that. You know, that's not going to tell me anything very important. That's not going to actually help me. And then as you increase your distance, the percentage of connections to the target area, which remains here, okay? At the long distance, the percentage goes from 100% down to a very low percentage. In other words, the long distance connections, the density is very low. So if I tell you, well, area V2 projects area 10, which is in the rostral pole, That's actually a very unlikely connection. It actually exists, and it might well mean something. It might say, oh, yes, and you get your back of your envelope out and work out some theory about how the brain works because that connection exists. It's, at this distance, it's in itself a rarity. It tells you something. But at the very short distance, it does not. And so you might think from this point of view, from the binary point of view, connected, not connected, the connections over long distance might be very informative, might be very important in explaining what the cortex is actually doing, but at short distances they're not.
I'm uncomfortable with that inference. I mean, it's unsurprising in the context of the empirical data, but if you thought about how you build brains in advance, for example, it's not a logical necessity that you be connected to your immediate neighbors. I think it does tell us something about the architecture, maybe about topography or something like that. Like I don't think it's a trivial or necessary finding.
Absolutely not. I'm saying that if I were to inform somebody in this room that two areas adjacent to each other are connected, that would not help them understand, because that is actually a pattern of the brain. You're absolutely right. It's a very distinctive pattern, and I'm going to come back to that as a major argument. This is how we can understand how the brain is actually connected. It's absolutely not trivial, but it's uninformative because that is the basic pattern. Wherever you go, neighboring areas are interconnected. That's the fact. And long-distance connections are actually really rather rare. Before you ask another question, let me push on because I might have a problem with time. So, okay, what's your other question?
That's just hasn't been the culture here. I was just going to clarify in one sentence. What I would say is it's uninformative about where you are in the brain, but it's not uninformative about how the brain operates.
Okay, so the huge bulk of connections actually is within a cortical area. If you look at the number of connections within a cortical area, it accounts for between 80 and 90% of the connections in the brain. So actually, and those connections within an area are within millimeters of their target. So the connectivity, the major connectivity, is what people refer to as local connectivity. You know, this hierarchical, this model of connections between layers, for example, in different areas. So that local connectivity is actually the machinery that we're talking about. The connections between areas represent less than 20% of the total connections. And 3/4 of those connections between areas are to the adjacent cortical area, or the one or two cortical areas. So the long-distance connection represents a tiny, 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 tiny fraction. Now, what I'm going to be interested here is to pose the question of what is the information content, what can be gained in terms of understanding the strength of connections between areas, and what is it going to tell us about the overall architecture. The point that you're making is perfectly true. What actually comes out of this, which was the first title I put on the board, was that there's a distance strength relationship which is actually very, very, very consistent. And that relationship enables you to make many predictions about the geometry of the cortex, about the way the areas are interconnected. And this is what I'm going to go over. How's the time going?
We're doing just great.
Well, what's that mean?
That means you're roughly halfway. We're about halfway.
About halfway,
Yeah.
okay.
But for clarification, Henry, so the distribution you gave us earlier, at this point, you would say this is invariant across the cortical sheet. We don't have to debate details. This is it, wherever you look, right? This is the point.
Yeah, yeah. It's a strong claim. I mean, our data is not. I need more funding to actually prove that. So,
I'll buy you lunch.
How do we measure the strength of connections? We make this injection. Oops. We make the injection here. We have these labeled neurons. So if we're looking at area V1 and we make the injection, we're going to have labeled neurons across all of these areas. These are the areas which actually project to area V1. This is the frontal eye field. It's composed of two parts. There's 8L and 8M. And if we want to know the relative strength of 8L, we measure the fraction of labeled neurons which is located in this area, and we express it as a fraction of the total over the total number of neurons which are labeled outside of the area that we're interested in. Outside of the area we're interested in is simply because there's so many labeled neurons in the area that we're interested in that, you know, it's a lot of work making all those counts. So it's just a small issue. So the strength of this area is the total number of neurons in area 8L, total number of neurons. I don't know if I have time to talk about why we go to such lengths, divided by the total number of neurons that are back-labeled in the whole of the cortex. And it gives you this measure of strength index, which we call FLN, fraction of labeled neurons, and it's 0.0214. That's the way we identify the different strengths of connections. Now, when we do that, there's a number of regularities. And the primary regularity that I'm going to be talking about here is the fact that as you go from a distance from the injection site, so there's 29 injection sites, it's actually more like 35 now, but there's 29 injections. We're summing up the labeling from one injection to the other, and we're finding that there's a progressive decrease in the strength of the connections as you march away from the injection site. This is millimeters in distance. And this decrease in the strength of the connection with distance has a lambda value. This is the space constant, which is around about 0.088. Actually, that's wrong. I've got a wrong slide here. It's more like 0.2, but bear with me. Last time I gave this talk, that set up a whole problem. Now, we're going to use this regularity in the strength of the connection to construct random networks using that as a probability, using that measure of decrease in density as a probability of connection existing. We don't have to take any notice of this, but this is an interesting observation that we made, which is that there's a log normal distribution in weights. This is all of the connections. This is a log scale with another log scale here, and you have this log normal distribution. And this is simply saying that there's a Gaussian distribution of distances, but we don't have to really, at the moment, rely on this. When we make these random networks using this exponential distance rule, which simply says that the probability of a connection is very high at short distances and is very low at long distances. So if we now take a number of nodes, which corresponds to the full network that we observe in the cortex, and we're going to build these networks and then ask ourselves, if we apply this exponential distance rule, how similar is the networks that we're building going to look with respect to the networks that we can observe in the data? So a characteristic feature of networks is to look at the subgraphs or what Alain referred to as the motifs. Look at the different motifs. So what you do here is you take all the different combinations of free. You can do motifs with 2, in which case if it's 2, it's any 2 areas which are not connected, any 2 areas which are connected with a one-way route going from A to B, any 2 areas with B to A, and any 2 areas with A to B and B to A. Those are all the ways you can connect 2 nodes. If you go to free nodes, the numbers get a little higher. Actually, there's 13 different ways. These are the 13 different ways you can connect free nodes. So what you do is you look at all the combinations of free nodes that you have in your network, and you look at the frequency of the distribution. And the argument that Alain was making, this is back in 2004, is that the relative abundance of these different motifs is going to be related to the processing that the network in question is involved in. So he was looking at power grids, he was looking at nervous systems, he was looking at social networks. This was the sort of argument this was part of the interest. I'm not using it from that point of view. What we're using it is simply to say that the motif distribution is characteristic of a given network. So this is the the 13, actually 16 different motifs, and so they're aligned along here. And if you look at the bottom here, we have the data, which is black. It's not actually that easy to see. So what you see is this is a completely connected triangle, which is actually is the most frequently observed motif that you get in the data. And there's a number of motifs which seem to be very actively avoided by the data. For example, I don't know, you probably can't see it, but a situation where A goes to B and B goes to C and C goes to A and nothing comes back again. This is a sort of cycle, and brains and nervous systems avoid that, and that is here. You see? Tiny. Brains hate that. Now, they do. They hate it. And the reason why I can say they hate it is you can take a network, the same number of nodes that we have in the data, and you connect them not randomly, but according to a constant distance rule. You're not giving a preference to nearby neighbors. And this is what you get. So if you were to talk about the nervous system as a sort of chance structure, this is the number of cycles that you would actually expect to get. This is the black dot. Can you see this at the back? There's a very little tiny black dot. So cycles are hated. Now the red lines are the predictions. No, we make many networks using the same number of nodes, same number of edges, and using this exponential distance rule. And these are the distributions of motifs that we observe. So the red lines and the black lines agree rather well. The blue lines agree very, very badly. So this is part of the argument saying that this weight-strength-distance relationship is actually useful for making predictions about the
But Henry, for clarification, this is only true when you limit the distance at which you're looking at that system, right? Because, for instance, if it would all be local, you should find all possible patterns because they have 100% connectivity, as you showed earlier. So these templates only emerge when you impose some spatial constraint, isn't it?
No. Actually, there's a tendency to get this wrong, which comes back actually to, when you look at this, there's a problem with this is a diagram taken from Watts and Strugatz. This looks like we're talking about local spatial connections. That's actually not what they're talking about. There's no notion of space here. There's no notion of space, and there's no notion of weight. It's a purely binary network. The small world network is a binary network. You don't talk about the strengths of those connections. In the original Milgram study, when people were sending envelopes, it wasn't whether, this man was his wife or his mistress or somebody he met in the tube or what have you. That doesn't come into it. You know, you don't know. And it's not to do with whether they're in Washington, D.C. or whether they live at the bottom of the street. No space, no weight. Purely binary networks. So this is where we are at the moment. There's no space here, but there is weight, and that's important. Now, there are other measures which are really very interesting, and I should have maybe put it in, but I'm used to talking over a shorter space of time. But there are other things that you can predict which I think are interesting. Let's come back to this, the rich club. So I made the point that with a very high density, it's really not very useful to talk about small worlds because everybody knows each other, so it's no longer surprising that there's a connection. And so in some sense, it's, the high density makes the small world notion just uninteresting and inappropriate in that sense. But I also talked about the idea of the hubs. So the reason why I was knocking on this notion of hubs is I think this idea that the network people have where you route information from one place to the other sort of ignores the fact that the nervous system doesn't send a message to here and then it gets routed over there. It doesn't work like that. There's a axon potentials come along an axon. They then synapse onto a cell. There's an integration of many axons coming onto that cell. The cell does something, and it itself projects to a number of neurons, and it sends its own action potentials across. So there's no routing of information. There's no single package that sort of wanders up from from the eye to the lateral geniculate to the area view 1, and then kind of crawls out here, and you get here. And that instruction is, hit this nut with the stone. It doesn't work like that. It's a very, very integrative process. So the idea of hubs is still is very much out there. There's a lot of papers on hubs. And the point is that with the densities we're reporting, if you try to do the maths to see if the hubs are more interconnected than they should be, you can't actually normalize. So you can't make that statement very clearly. So what there's a whole number of people working on this, so I'll go through that at the end. We've looked at the number of clicks that you can find. So a click is a subgraph within a network with 100% connectivity. A click is a group of people where all the members of that group interact with each other. So clicks have a little bit the same function that we were talking about earlier on. It turns out that when you look at the click size in the data, which is these 29 areas, we find 10 clicks. We find 11 clicks, and these clicks are the number of areas that have these 100% connectivity. This is in the data. When we use the when we use this exponential distance rule, we can make a fairly good prediction of that number of clicks. So the argument is that we're saying is that the number of clicks, or the click structure, and these clicks go across areas. So this is color-coded. It's prefrontal, which makes a major contribution to the clicks: frontal, parietal, and temporal. These are the areas which are participating in these clicks. So if we increase the number of injections, we're going to increase the number of, we can increase the number of members, but the clicks that we found and discovered will remain the same. The probability of finding these clicks by chance is vanishingly tiny. It's a very, very low number. And the capacity of the exponential distance rule to capture these clicks is really rather good. So there is a click structure which is in itself rather remarkable. It could be certainly involved in cognitive architectures, like Damasio has proposed this divergence-convergence architecture, which would be important for multisensory integration, and it could be involved in that. If you look at the Dehen and Changeux, they have this workspace which they publish every year. It's a very robust project, and so the kind of click structure could come into that. It's something which I think could have an important organizing principle.
Yet, but I don't agree with that.
With what?
That the idea of a global workspace would fit your click idea.
Okay.
Because the notion of a global workspace is that you dynamically couple chunks of cortex together, right? So it's a dynamic concept. Well, what you're describing here is really a structural concept.
Yeah.
So I think these are two different.
Sure.
Function versus structure again.
Yeah. No, but that's not a problem. I'm not saying this is a global workspace. I'm saying it could be. You could be interested in this click structure in terms of understanding the underpinnings of global workspace. But that's true, Paul, for any observation of structure. I can be showing you a synaptic cleft and saying, well, this is the structure of my synaptic cleft, and this is, and you can say, but Henry, this is not telling me about how vesicles actually go across the cleft and how the exon potential is.
No, that was not the point. I think we can discuss it later. Okay, I don't want to distract you too much, but I think it's at the core of the workspace idea that you really continuously dynamically couple any arbitrary bit of cortex with another arbitrary bit of cortex, and that then forms the conscious scene. So the notion is the flexibility of this dynamic linking, while the click idea really gives you a very fixed, if you want, structural backbone structure of cortical structural architecture. And it might support then global workspace, but it doesn't automatically imply a global workspace. That's what I'm trying to say.
No, that's fine. I have no problem with that. So why am I referring to the brain in space? The small world network is a binary, it's a binary network which deals with with structures being connected or not connected, and it has no notion of space. What we're claiming here is that if you look at the strength of connections and you look at the distance of the connections, you find that there's a very strong relationship between those two, and that is what enables you to make predictions about interrelationships between the cortical areas that you haven't directly measured or used. And when you make networks using that principle of strength and distance, it turns out that the features that you can observe in these artificial networks map on very well to the features that you can see in the actual cortical data. That's why we're calling this the brain in space. And I think what we need to do now is to have much more information about that kind of detail and understand that the brain is actually confined. There's a constraint, it's a spatial constraint, and that there's these strength of connections which are going to be playing a role at different distances. So that's the meaning of the title. Now, this is a flat map so I'm going to switch over now. This is a flat map of David Van Essen. And this is area V1, and this is area V2, which normally wraps around area V2. And the point I want to make here is that I've given you evidence that there's a very strong relationship between the strength of a connection and the distance over which that connection is. So if I look at a neuron sitting in area V1 here, and I look at the range of strengths of connections which I would predict it would be having with the cortical areas which are around it, and I look at an area V2 neuron out here, and I look at the areas that it should be connected to, you would make a very simple prediction. And that is that the strength-distance relationship of this neuron should be very different from the strength-distance relationship of that neuron. And it turns out that's true. So what we've been doing is, and I'm not going to show you data on this, but what we've been doing is looking at doing an injection in central V1, V2, and V4, and summing up the strength of connections. And it turns out that if you do that, in central V1, V2, and V4, you sum up the strength of connections, and they're very interconnected with the ventral stream areas, which are all down here. If you do the same thing for the far periphery, they're strongly connected to the dorsal stream areas. And it's a very, very radical difference. So it turns out that you could actually think of ventral stream. Do you all feel comfortable with ventral stream and dorsal stream? Yeah? Hands up. Somebody here must not know it. I mean, you're robot people, right?
Half of them won't know it.
Yeah, okay. So the dorsal stream, John Cass here is a specialist, so it's a bit embarrassing. The dorsal stream is coming out of the visual cortex, and it's going to parietal areas, and it's much more to do with space, orientating the body in space and understanding space. The ventral stream is coming from the visual areas, it's going down to the temporal pole towards the hippocampus, and it's much more to do with object recognition. So people working on the visual system have been very influenced by these dorsal and ventral streams, and the understanding is that they have very, very different finalities. What I'm saying here is that if you look at the structure of the visual system, and if you believe the first part of the talk, you would make a very simple prediction. You would predict that the dorsal stream will be more heavily interconnected with the peripheral visual system, and the ventral stream would be more heavily interconnected with the ventral system. And it turns out that when...
With central, I guess.
Sorry?
You were saying dorsal peripheral, so ventral would be more central.
Yeah.
Okay.
And so we've done the same sort of analysis which I've been talking about earlier on, making the FLN and distance and what have you, and that turns out to be true. So I think that something which I didn't tell you in the last thing, if you think about this distance-weight relationship, it tells you that you would you would make it a very straightforward prediction from that. You would predict that the areas of the cortex are optimally placed to be next to the areas that they're most connected to. That's called optimal placement. And you can test that by moving areas about on the brain and seeing if you can reduce the amount of wire. We did that. And we found within the monkey that you can't reduce the amount of wire. There's no way you can reposition areas and reduce the amount of wire. And this idea of wire minimization comes back a long way. Many people have discussed this notion as being a sort of central design feature of the cortex, that the minimization of wire is actually a large part of what's going on. And our result in the monkey says that's absolutely the case. There's no way. There's a paper by Kaiser and Hilgertag, which is very, very strange in PLOS Biology, saying exactly the opposite. But other than that, it's been hugely subscribed to. Wire minimization. What I'm saying here now is that there's something about the shape of cortical areas which is very enticing, because if our results in the visual system are at all indicative, it would suggest that the shape of the area optimizes the connectivity of different parts of that area with respect to the areas which are around it. And I think that that's an idea that comes out of the wire minimization, so you have an optimal placement of the cortical areas, but you have an optimality in the shapes of the cortical areas. I think this gentleman was first.
I was just going to ask, is that taking into account soul sign gyri, or is that for the flat inference, the wire minimalization?
I'll answer that in a second. Let's hear what Leah's question is going to be.
If you go with the wire minimization, then the absolute size of the neocortex would change how brains process information. So in a small brain, frontal cortex is closer to V1.
Yes.
And if you're selecting for absolute wire length and trying to minimize that, that would say that the macaque monkey's fine, but it's going to be hard to extrapolate to the human, which has a much larger neocortex.
Okay.
So you're saying you have a lot more local processing in larger brains and more global processing in smaller brains using the same wire minimization rules, correct?
Well, I think the two questions are actually very related. So we've done exactly the same thing in the mouse. When you do the mouse, you have 15% possibilities of reducing the wire by moving the areas around. And I wanted to talk to John and Leah about how they would sort of see that, because it's, for us, all very sort of surprising. And you can take your monkey and you can, instead of measuring the distances I was talking about earlier on, are distances through the white matter. So we're claiming that they're going along the trajectory of the neuron. You can do the same thing by taking surface measurements. When you take the surface measurement, you're sort of unfolding that brain. And it changes the way the distances are related. And it changes the weight-distance relationships. And what we're working at the moment is to see how this folding comes into it. Because it's giving you a much more, you remember I said the distances are Gaussian? Well, if you remove the folding, the distances become much more broader. And you lose that high precision, that high number of distances over a very small mean. So I think that the two questions are very interesting, and I think they're related. And actually, we have an eye and air with John. He's a silent partner in the sense he doesn't get any money, but we're kind of interested in not only comparing macaque with mouse, and we're in the middle of that at the moment, but then also comparing macaque with microcebus, because then you stay within the same species, but you have a smaller brain. So it comes back to that question there.
How much of the variance does the weight-distance relationships account for? And then the follow-up is, for the part that it doesn't account for, how critical is it? So I'm thinking in part of the Human Brain Project, where the idea is to take a predicted brain and use that predicted brain as a model. And to the extent that you can use a weight-distance relationship as a good predictor, that means you're in good shape. But if it turns out that the 22% that you don't predict is really important, then maybe you're not in such good shape.
The 22% comes from?
I made up the number. I'm asking what is the proportion of the variance of connectivity that you get by making this distance prediction? So you're saying it's a very good predictor, but you haven't really been quantitative about how good
Ah, okay.
So, I mean, is it predicting 100% of the variance? Well, obviously it's not from your own data. It's kind of clear that it can't be because there's so much conductivity. But, well, actually, I retract that sentence. But you can think of it as a statistical measure. I'm predicting 50% of the variance, 100% of the variance. I'm guessing it's somewhere in between there, but you haven't said, and I'd like to know so I can think about how effective it might be for, say, building a whole brain simulation if you use this on as a constraint on that simulation.
Okay, where it doesn't work out very well, it's good at making these predictions about motifs and clicks and things like that. Where it works out not so well is if you reverse it and you try to use the exponential distance rule to predict the range of strengths, then you fall into a problem. It doesn't seem to do that. And Xiaojing Wang and his group are now having a different approach. They're looking at the similarity of connection profiles, the fingerprints, if you were, and by using generative models based on that, incorporating this exponential distance rule, they're able to get much better predictions about the ranges of strengths. So it's a bit of a vague answer to a precise question, but there's other ways around it. So why minimization? So coming back to these neurophysiologists and what they're interested in, this actually isn't. I haven't got a slide of it, but this is not Feldman and Van Essen's model. But it's just I've just come to it quickly to remind you about what is this whole issue of hierarchy. So Feldman and Van Essen did a very famous model, and I haven't got it here. I'm sorry, but you would all. It's been shown many, many times, and people show it for a number of reasons. They sometimes show it to frighten people. You know, just shut up, don't talk, because it's kind of complicated. And basically you have many more lines than this, and it goes up to area 46, which some people think does everything. And so it starts off with area V1, so you march out from so this is a simplified model. You go from V1 to this is a simplified Feldman and Van Essen. Very, 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 very simplified. You go from V1 to V2 to V3, and you end up in prefrontal cortex. So basically what they're doing is they're taking the observations of Pandua and Kathy Rockland and many, many people who published before 1991 who found very strong laminar patterns in the forms of connections. So if you look at the connection going from V1 to V2, V1 is the primary visual area it receives from the lateral geniculate, and V2 receives its information from V1. It doesn't receive it, not principally from the lateral geniculate, and then it goes to V2 to V3 to V4 to V5, and you have the same thing in the auditory system and the somatosensory system. So you have these primary areas which get their input from relay nuclei in the thalamus, and then they go to subsequent areas. This is the notion of hierarchy. And it's thought of as being hierarchical because if you silence V1, then the response in V2 disappears. And if you silence V2, the response in V1 is relatively unaffected. The mystery comes when you look at the number of connections. So this is felt to be true in broad terms in the system. When you look at the numbers of connections between the areas in these different layers, they actually turn out to have about the same number of connections going from V1 to V2 as going from V2 to V1. So the V1 to V2 connections are understood to be driving. Murray Sherman and many other people have talked about this. The projections from V2 to V1 are much more mysterious. And so when things get mysterious, they become modulatory. You modulate what's driving. And that's really rather a cop-out because we know that actually the thalamic input to V1 itself modulates activity going on in V1. There's a recurrent excitation in V1, which is very important. So this simplicity of modulating versus driving is not that easy to manipulate. But nevertheless, Jean Boulier and other people have shown that there is this relationship between the dependency of the higher areas on the lower areas. And the phenomena that I'm talking about at the moment is that there's a laminar distribution. So the input to the cortex comes into layer 4. The cortical layers go from 2 to 6. The upper layers do not have any projections to the lower subcortical structures. All those projections come out of the infragranular, that's to say the lower layers, okay? And the connections between cortical areas come out of the upper and lower layers. So when you're looking at this is probably going to be on a slide here somewhere. Oh. How am I doing for time?
15.
Well, I don't have the proper slide to go into this, so I'm probably overloading you with stuff here. But there is a strong laminar pattern which fits onto this driving versus modulatory to some extent. And this is what enabled Fellman and Van Essen to propose a much more complete hierarchy than what I'm showing you here. This is a sort of bare bones. We're choosing here the areas that we've been able to actually investigate. And then we're reorganizing the areas here and basically using this quantitative way of measuring exactly the number of neurons involved in connections. We've been able to quantify an index of hierarchy, and this enables us, with very few injections, to obtain a hierarchy which looks very much like the Fellman and Van Essen hierarchy, except for, here, this is the frontal eye field. The frontal eye field is a prefrontal area, which is important for directing the movement of the eyes and for directing attention. And in the Fellman and Van Essen model, the frontal eye field is right at the top of the hierarchy, and interestingly, in our hierarchy, the part of the frontal eye field which is involved with small saccades is at a level of about V4. And I think that's interesting. So but when we look at the pathways between all of the visual system, we are able to show that there's a very neat compartmentalization that you don't find in rodents, and you do find in the macaque, and you find it across the whole of the extrastriate cortex, and it's really quite striking. And it was actually noted by what's their name? That couple that were doing anatomy back in the 60s, Thingy Bob and Thingy Bob.
Just say a random name.
Okay.
We won't know.
It was noted. It was noted back in the 60s. What you find is that you have this short distance feedback connection. So this is a high level and this is a low level, and long distance feed forward connections coming out of layer 3, and a similar sort of organization there. And we've made something about that. But I think I'm probably losing you. I'm losing myself, actually. Okay, so these are the people that did the work, as they say. This is a remarkable chap who was able to keep all of these threads going at once over about 7 years he was in the lab. Zoltan Torocai, who is originally from Romania but now works in South Bend, this is his name here, is a network scientist, and this was his postdoc at the time, Mahak, who's now at this Arsgai Rafsas, who's now back in Romania. They were responsible for a lot of the all of the modeling of the network science that we were using. Ken Knobloch is not here. Strange. So the collaborator I work with intensely is not here. He should be here. He should have his photograph here. He's called Ken Knobloch and played a very important role. Colette played a rather minor role. David Van Essen collaborated on part on this in numerous ways, but enabling us to improve our imagery of connectivity. And these are the students that made all sorts of contributions who appear on publications to come and publications which have come out already, and I did a lot of the back work. So thank you very much for your attention.
All right. Now that we know how the brain works, let's connect.
So you didn't directly say, and probably it isn't true, but I'm going to sort of cartoonize your claim. The distance predicts hierarchy as well. I think that's probably not entirely true, but it's sort of roughly true. Bear that sentence out, and then you can comment on it. then it seems to me really interesting when you find cases that violate those generalities. So the frontal eye field case becomes really interesting. So I guess the more general thing that I'm trying to mull in my mind, and that maybe you can comment on, is the relation between the stuff that's predictive and then the rare exceptions, and what might be the potential power of those. So like, there's presumably a reason why frontal eye field, even though it's kind of far away, is actually playing a pivotal role.
Okay, yes, that's very, very true. So the frontal eye field is this prefrontal area that directs the movement of the eyes and also has a role in directing attention. And when we look at these hierarchical relationships in terms of the laminar distribution of neurons, turns out to be at a level that makes it look much more like an early visual area rather than this higher up area. And the point that you're making is that this is an anti-hierarchical connection, and it is also much stronger than you would predict. So it's stronger than you would predict. And in actual fact, we found 5 or 6 of these types of connections. What's actually remarkable, people get very sort of sly about Feldman and Van Essen's model, but what's actually strange is they got it so right. There's very, very few violations. There's very few connections which show the opposite, that don't have the correct relationship. I mean, the correct relationship for the frontal eye field would be a feedback modulatory type relationship with area V4. That's what it should have. What it actually has is a driving type, from the signature of its connections, a driving type projection to area V4, which is not what you would predict for these higher order areas. The higher order areas are meant to modulate the posterior areas, and the posterior areas are meant to drive the ones which are in front of them. And that's a simplistic thing. So with these 3 or 4, I think there's about 4, anti-hierarchical pathways, you can go from V4 to area 46, or area 10, and back again, only through feed-forward pathways. You don't need to take any feedback. Let's say Americans do this, yeah? It means, I don't know what it means.
We call them scare quotes.
Okay. Without using apparently modulatory, and you have a few very unusually strong connections that stand out, and yeah, that's, I think, a very interesting thing to look at those. It's on the agenda.
To come back to strength, so you gave me the definition now, how you actually define strength, so I'm happy with that. But my question now to follow up is, how much does it now relate to physiological impact? So would you see if you have, how much this correlation, how you define strength actually then, have you checked how the activity in one area would actually influence the other area?
Okay, so we're collaborating with a chap called Pascal Fries. And over the last 3 or 4 years, it's become clear that so Pascal Fries is well known for a number of publications making the claim that oscillatory coherence increases the communication between neurons and between areas. And a recent sort of touch on that is you have a distinction between gamma oscillations and slower oscillations in different layers of the cortex. So the upper part of the cortex dominates in feed-forward projections. I didn't have time to go into that. And the infragranular, the lower layers, predominate in the feedback. And from that alone, you would make a prediction that the gamma coherence would be much more important in the feed-forward pathways, and the slower oscillations would be more important in the feedback. So he's looked at that using Gainsborough causality relationships and has been able to construct a functional hierarchy which actually maps onto the structural hierarchy in a rather interesting fashion. But it's dynamic. And so what's interesting is you see areas flipping their position according to different positions of the task. And the other interesting thing is that when you look at those areas that flip, it turns out actually it's the frontal eye field, and it turns out that they flip, and you could also explain the way they're flipping by how much of their connectivity you're taking into account. If you take what I showed you in that hierarchy there, I was doing a little trick. It's the areas, it's the connections of a restricted group of neurons. If you took the whole of the connectivity, including the prefrontal cortex, that would drag the frontal eye fields up to a higher level. And I think that's governing this sort of dynamic flicking. But so there is, just another thing, I see two problems besides the Human Brain Project. There's another problem, and that problem is to do with the language we're using. The functional connectivity thing is an abomination. But the other problem is you now have millions of papers now which are saying, well, we can now do human connectivity, because we really want to understand the human brain, right? We can do human connectivity because we have diffusion MRI. Okay? So actually, if you look at the correlation, this is track tracing, if you want the jargon. What I've been showing you is track tracing. Very old-fashioned. You put a substance and you watch where it's dragged to, and then that gives you a pathway. If you look at the correlation between diffusion MRI and track tracing, it's very poor.
Moreover, you have no directionality.
No directionality.
So thank you for the talk.
No, wait, Yvonne. We have this gentleman before you.
Oh, I'm sorry. It's like I haven't seen.
You mentioned small brained animals once, and then you also, in responding to a question, but then you had your, where you said that 67% of the possible connections in macaques are actually found. Does that hold for small brained animals as well, or is it a greater proportion there? Do you know?
Well, so there's a recent paper of the Allen Brain Institute where they published in Nature. In Nature, they published this mouse connectome study. They're using antegrade, and they're reporting a much lower density. And so what we've been modeling at the moment is the Allen Brain Institute studies. The first author is somebody called O.H., and they're reporting a much lower density. And the much lower density and the others, so you remember I mentioned that there's a 15% economy of wire that you can do by moving the areas around. So if I had the slide and I showed you, you remember I showed you a slide where you looked at the probability of being connected with distance, and it goes in the macaque from 100% to 0%. In the mouse, it's not like that at all. It goes from 70% to 40 or 45%. So it's a very much more flatter distribution, and there is an exponential distance rule, but it's much weaker. It's not specifying things so much. And I was wanting to ask people here if certain questions like that.
Well, isn't that surprising, given that there's a smaller number of areas? If anything, I'd expect it to be a higher percentage, or no?
No, the density is lower, so you could think the density would be higher, but they're using a different technology from us, so that could be a part of the problem. John?
Yeah, just a quick question. Given the anatomical structure that you depicted, can you infer something on the temporal dynamics? Like, can you hypothesize something on temporal dynamics that you would expect out of such a network?
Well, I'm collaborating with people who are looking at that. It's rather sensitive, so I'm not going to go into it. But yes, you can. There's measurements you can take which will enable you to make kind of combine the data that we have with measurements of spine counts and things like that, which you can sort of argue gives you some sort of way of modeling the dynamics of it. But I'm not going to talk about that. I think Johnson.
Go for it.
I'm just wondering about comparing mouse with macaque monkey. Part of the problem in the comparison in both cases would be in defining cortical areas. And you're using more cortical areas in a macaque than Van Issen and Fellman did originally.
Yeah.
And the number keeps going up.
Uh-huh.
I don't know what the ultimate number will be. But, and some of us, me being one, feel that the number of proposed areas for a mouse is too large. And it's too small for a macaque and too large for a mouse. And this would have some influence on how you come to the connection area conclusions.
Yeah, absolutely. I think that's a very important point. What I'd say in reply to that, you remember I said that you had this log normal distribution of weights. it's rather interesting because you go to the single cell level, and the synaptic weights have been reported as also being log normal. So if you were to get more and more areas, you just say, well, I want to have a central visual area and a peripheral visual area, and I'm going to divide. You could go on and on dividing the brain. I think that you, at least in terms of that log normal distribution of weights, and there are distance properties as well at the single neuron level. I'm wondering if, as you get it finer and finer, and you'll be talking about columns, you wouldn't necessarily be talking about areas anymore, and then you'll be talking about maybe the single neuron. Obviously, your density is going to plummet, so you're going to have you can then say, I have a small world at the single neuron level, very probably. You'll have a very, very large number of nodes, and you'll have reasonably short hop lengths, and you'll have a high cluster. Well, we don't quite know about the clustering that well, but you'll have a higher clustering than you might think. So, yes, the number of areas. We're using 91 areas in the macaque. David Van Essen thinks that 130 would be the sort of top number of areas in a macaque. That's what he's suggesting. So the density difference between mouse and macaque could be influenced by us using less subdivisions per size of the area, if you will. But interestingly, despite that, when you look at the mouse data, there's a surprising lack of connections between areas which are not that far apart, which would not be the case in the monkey if you compare them.
So, can I ask a question? So, it'll be really interesting to see the microcebus, because if the microcebus is like a Galago, MT is bumping almost right up against V2. So let's just use the V1 MT connection as an important functional connection.
Mm-hmm.
I'm sorry I said that, but anatomical connections that we know exist in all primates. But MT is relatively close to V1 and V2 in some primates, and then it's moving further and further away in macaque monkeys. So what happens to the close areas are more densely interconnected, where we do expect MT to have fewer connections in a macaque than it does in a microcebus, because in microcebus it's much closer.
Right. Well, I mean, MT in the macaque, maybe I've got this wrong, but I think MT in that region is the only area getting a direct input from V1, right? The macaque. It's got a belt of cortex which is V1-3, which is it's very striking, and the input to V1, the input to MT is, it's got these neurons in 4B and a lot of upper layer neurons. It's a kind of impressive. It's an impressive input to an area which is so far away. So the prediction would be, the distance rule would say that the connectivity of MT in the smaller animal would be denser, because you go into an a-dimensional way of looking at the brain, and that's what we're doing, comparing the mouse and the macaque. But one would predict that it would have a stronger connection, but that would be, yeah. But then there are outliers, and so it might turn out that MT is an outlier in some way in the macaque.
I mean, the reason I get it is I think there's a notion that areas in evolution are added hierarchically. First there was V1, then there was V2, then there was V3.
Right.
That's not how it works.
Mm-hmm. Mm-hmm.
What I said is that there's this general feeling, I think, in public, because they like to think of hierarchies, that in evolution, areas are added hierarchically. First there was V1, then there was V2, then there was V3, V4, and so on and so forth. But in fact, areas are interspersed between other areas. I would argue that probably MT is an older area, and
right.
some of these other areas are interspersed,
yes,
or
yeah,
merging between known
right,
areas, and so it makes it really hard to make comparisons across species with these. Although I love the quantification, it makes it more difficult.
Oh yeah, yeah,
We have one more question here, then.
Yeah, my question is about the size of areas, and if you observe any kind of like bottleneck within the hierarchy. Do you observe that, like in the middle of the hierarchy, starting at the sensory areas, is there any shrink in the size of areas that would then expand and account for any process of convergence or bottleneck that is very trendy at the moment in
bottlenecks are trendy
in machine learning? Yes.
Oh, okay.
Also in the local bars.
Local bars.
Yeah.
Okay. I can't answer that question. It's true that area V1 and V2 are huge. They're very, very large. That's true. And as you, at the early stages, as you go up, the areas get smaller. That's kind of true. But then when you get to the top, at area 46, so really, the elephant in the room is that we talk about areas, as John points out, glibly. There's no single definition of Courtauld area, really. It's very easy when you're talking about V1 because you have a very clear border and the physiology and the anatomy map on very precisely. That's not always the case. So but yes, so if you were to take your point, area 46 is at the top of Perlman and Van Essen's hierarchy. It's very big. And V1 is at the bottom, and that's the biggest area in the brain. So it would say you had an hourglass. You have a bottleneck.
Okay, great. All right, Henry, thank you very much.
Thank you.
Great talk.